All right. So Tuesday, we covered all of this stuff about the little ballet that the chromosomes undergo. And when this was discovered in the late 1800s, it was not immediately clear what was going on. It seemed like it was important, but the chromosomes were these little objects that turn up under the microscope that by themselves are transparent, but they tend to take up color very strongly when you stain a slide with one of these new dyes that were being brought out. And they do this funny little dance when a cell is about to divide, and we call that mitosis. They do they appear and they clump together, and then they line up in the middle, and then they shout, "Hey, Macarena!" Actually, I'm kidding about that part. And then they separate at the centromere, and separate chromatids pull apart to opposite sides of the cell, and then two new cells appear. That's mitosis. We talked about that at great length. And then there's the second type of cell division called meiosis, which is used at least in you and in animals only to make egg cells and sperm cells. Plants, fungi, and protists, it can get a little weird, and we probably won't have time to cover that. But at least in your cells, it's only used to make your eggs or sperm. And it's a two-step division, each of which is divided into prophase, metaphase, anaphase, and telophase. And you end up with 4 cells, each of which contains one of each chromosome, and each chromosome consisting of one chromatid. The reason you ought to care about this, other than me giving you a bunch of stuff to memorize and spit back on the second exam on November 4th, the reason this actually matters is that we're starting to get to where we can see that we've been working on opposite ends of the same jigsaw puzzle. Mendel worked out that genes come in pairs. Every organism inherits one gene of each pair from each parent and passes on one of each pair to each of its offspring. Meiosis shows that you pass on one of each of your chromosomes to each offspring, and you got one of each chromosomes from each of your parents. You have 2 copies of a large chromosome that we just call chromosome number 1, and you got one from mom and one from dad. You've got 2 copies of the second to largest chromosome, which we just call chromosome 2, and you got one from mom and you got one from dad. And you'll pass on one of each of these pairs to any offspring that you have. And this somehow has to be important because we know that if an egg or a sperm ends up with a chromosome that's missing or an extra chromosome because of a mistake in meiosis, that can lead to fertilized eggs that don't develop or that don't develop properly. So genes and chromosomes are actually following the same rules. Mendel worked it out by counting a bunch of bleeping pea plants. Microscopists of the late 19th century worked it out by staining a lot of slides. And yet here we are, these are following the same basic rules. So is there some kind of link between them? Back by 1902, a couple of people named Sutton and Bovary had independently of each other proposed that there was a link, that genes were physically carried on chromosomes. Whatever these genes are, they are physically parts of, or carried on, as we often say, physically parts of these chromosomes. Genes and chromosomes cannot be the same thing. Pea plants, for example, only have 7 pairs of chromosomes, but they've got a lot more than 7 genes. So genes are not identical with chromosomes, but genes must be physically located somehow on chromosomes. That stirred the interest of a professor who at the time was at Columbia University in New York City. Although he came from down south, he's still the only Nobel Prize winner to have graduated from University of Kentucky. So go SEC. His name was Thomas Hunt Morgan. And he and his students started studying genetics. Instead of using pea plants, they used common fruit flies, Drosophila melanogaster. If you have ever left your bananas out in the kitchen for a little bit too long, you might have ended up with Drosophila melanogaster. These guys are really small, but they feed on overripe fruit, and it's easy to get an infestation of them if you leave fruit out until it starts getting brown and soft. So some of you may have had to deal with these little guys before. Why fruit flies? Well, they are very cheap to raise. You can raise them on just fruit that the vendor is about to throw away because it's gotten brown and soft. They don't take up much space. These things are maybe 1.5 millimeters long. So they're not very big. You can raise them. They used to use glass milk bottles back in the day. And they breed fast. It takes 7 days to go from a freshly laid fruit fly egg to a fully physically mature adult. And they also have a lot of traits that follow simple Mendelian rules. They vary in body color, shape of the bristles, shape of the wings, eye color. They've got lots of traits that are fairly easy to see. And once you work out tricks as to how to do it, they're very easy to handle. So they make a convenient system for studying inheritance. You know, as I've said, Mendelian genetics applies to virtually all eukaryotes, but it's much, much easier to work with fruit flies than it would be to work with, let's say, elephants, which cost a lot more, need much more space, and will kill you and step on you if you don't treat them right. Fruit flies are far easier to handle. They've also got 4 pairs of chromosomes, and these are what they look like. In certain cells, Drosophila chromosomes happen to grow unusually large and thick. If you look very carefully, you can see banding patterns on these. They look like they're stripy, and that makes them also fairly easy to study. And so fruit flies are a great example of what we call a model organism. We could study Mendelian genetics in anything we wanted to, but practically it gets expensive and very difficult if we wanted to study it in elephants, even though Mendel's laws apply to elephants too. And it's much more handy to study fruit flies. You can raise lots of them. It's very cheap. Some of our classes in this department used to do it. And that's why, even though there are politicians like Sarah Palin who disapprove, fruit flies are used in fundamental genetics research all over the world. They still are. There's still lots of people that study biological principles using fruit flies as a model organism. And in 1909, one of Morgan's grad students, a guy named Calvin Bridges, discovered what we now call a mutation. He discovered a mutant fly. This particular mutant fly had eyes that could shoot optic blast laser beams, so he always had to wear a visor. Actually, I'm kidding. This was not one of the X-Men mutant flies, but a mutation is just a case in which an allele changes within a single generation. We will talk later as to exactly what's going on once we talk about what genes really are. But a mutation is basically the appearance of a new allele, seemingly out of nowhere. And this particular one was a fruit fly that had white eyes instead of the normal red eyes. So that's a normal red-eyed fruit fly on the left and a white-eyed fruit fly on the right. Well, Bridges took that white-eyed fruit fly and crossbred it with red-eyed fruit flies. And if you cross a purebred red-eyed fly with a white-eyed fly, the offspring all have red eyes. So red eye color is dominant to white. That's the same logic that Mendel used when he worked out that purple pea color was dominant to white. By the way, for a lot of organisms, we can define something called the wild type. And in fruit flies, that means if you were to go out into, well, according to a former student, if you were to go into the produce section of Harp's grocery store and catch a lot of fruit flies, because that seems to be a place where they like to hang out, or at least I was told that once because I had a student that worked there, you would catch a lot of wild fruit flies and they would all have red eyes. You almost never see white-eyed fruit flies in the wild. And that being the case, we can define red eye color as what we call wild type, which just means the phenotype that we virtually always see in nature. So if I talk about wild type fruit flies, I just mean fruit flies with red eye color. Why I needed to introduce that topic, you'll see in a bit. So if we cross two hybrid flies from the F1 generation, two of the offspring of a red-eyed fly and a white-eyed fly, you get 3 red-eyed flies for every 1 white-eyed fly, and that's exactly what Mendel's laws would predict. So far, this looks exactly like crossing purple-flowered peas and white-flowered peas. However, Bridges noticed something was a little off, because when he did this and he looked at the flies in the F2 generation, all of his red-eyed fruit flies turned out to be males. And that's another advantage of fruit flies. With a little practice, it's fairly easy to tell the males and the females apart. You need a microscope, and it's not super obvious, but with a little practice, it's pretty easy to sex fruit flies. And all of his white-eyed fruit flies in the F2 generation turned out to be males. In fact, in the F2 generation, half the males were red-eyed and half the males were white-eyed. None of the females were ever white-eyed. Huh. The only way you could get a white-eyed female was to cross a white-eyed male with one of those F1 females, in which case a quarter of the offspring would be white-eyed females. But if you mated one of those white-eyed females with a red-eyed male, all you got was red-eyed females and white-eyed males. Huh, what's going on here? There's this odd link between eye color in fruit flies and the sex of fruit flies. Okay, sure. We have a student who had to leave and is rejoining on laptop. Okay, there we go. We have readmitted our student, and let us move on. Okay, here's what is going on. I said that in cells that are diploid, which in your case is all of your cells except for your eggs or sperm, chromosomes come in pairs, and you get one from each parent, and you will pass on one of each pair to each offspring. That is almost true. it turns out that in many organisms, there's one pair of the chromosomes that's a little different, and it actually depends on the sex of the organisms that has them. And we call these the sex chromosomes. And normally, they determine the individual's biological sex. In most mammals, and by a happy coincidence, also in fruit flies, if you are biologically female, most of the time, there are interesting ways in which this can end up not quite working. But typically, if you're biologically female, you are born with 2 copies of a large chromosome that we call the X chromosome. If you are biologically male, what you normally get born with is 1 copy of the X chromosome and 1 tiny little chromosome that we call the Y chromosome. So these happen to be chromosomes from a female human. Remember that we pair up these chromosomes by use of the size and the position of the centromere and the banding pattern. So here you can clearly see that each of these chromosomes is kind of stripy. And this human female has 22 pairs of chromosomes that are not sex chromosomes. And by the way, a chromosome that isn't a sex chromosome happens to be called an autosome. And then down there, there is 1 pair of pretty large chromosomes called X chromosomes. Okay, these are chromosomes from a human male. And again, human males have 22 pairs of non-sex related chromosomes, 22 pairs of autosomes, which we just number from 1 to 22. But down here, males have 1 X chromosome and 1 dinky little thing called a Y chromosome. So the normal way of things is that human females, biological females, are born with 2 X chromosomes. Biological males are born with an X and a Y. And nobody is ever born with 2 Y chromosomes. There's 2 reasons for that. One is that the Y chromosome is really small, and it doesn't have, I'm getting ahead of myself, but it doesn't have many genes on it. The X chromosome carries a lot of genes on it that everybody needs to survive. You cannot live if you don't have at least 1 X. Okay, I'm getting a little ahead of myself, but let me get back to that. I will explain this in just a couple of slides, but suffice it to say that nobody ever has 2 Y chromosomes. Why people can't exist. Morgan had actually gotten his idea to study fruit flies from a fellow researcher at his previous workplace. He taught at a women's college in Pennsylvania called Bryn Mawr, which is still there, by the way, and very, very good, I am told. So he'd gotten the idea from his colleague at Bryn Mawr before Columbia hired him. This is Nettie Stevens. I finally get to talk about women scientists because by the time we get to around the beginning of the 20th century, they're actually starting to let women study science and all that. Oh gosh. And she'd made the discovery that many insects have chromosomal sex determination, and it works out in the same way that it does in humans. Females have 2 X chromosomes and males have an X and a Y. This isn't universal in the animal kingdom, by the way. With birds, it's actually reversed. With birds, females are XY and males are XX. Except just to be difficult in birds, that's called being WW and WZ. Just because we have to be annoying about that. In alligators and in turtles, your sex is determined by the temperature at which your egg incubates. If you incubate your eggs a little bit cooler, I think they end up as females. A little bit warmer, they end up as males. I think I could have that backwards, but it's not critical here. Quite a lot of fish are all start as one sex and then switch over to the other. Or there's some that are, we call this hermaphrodites. They have both sex organs at once. So sex determination is all over the map. There's nothing universal about it, but in mammals and in fruit flies, normal females have 2 X chromosomes, males have an X and a Y. By 1911, Morgan had put the pieces together and realized that you could explain this pattern that he was seeing in the inheritance of eye color in fruit flies by assuming that genes are physically carried on specific chromosomes. And specifically, the gene for eye color is physically located on the X chromosome. And that is something we call sex linkage. How's it work? Okay. If you are a female fruit fly, you have 2 copies of the X. If you are a male fruit fly, you have 1 X chromosome and 1 Y. We write sex-linked alleles by writing an X or occasionally a Y, but in this case, it's only an X, and adding a little superscript. And wild type we usually represent with a plus sign. So this X plus up here means a copy of the X chromosome that is carrying a wild type allele for eye color. Part of the reason I'm doing it this way is because X with a capital W and X with a lowercase w would be very hard to tell apart. So we're using the plus here to mean wild type. And in this context, that means red eyes. So the female in the very first cross that we talked about was a red-eyed female that never had any white-eyed ancestry. So that female is homozygous. She's got 2 X chromosomes because she's a female, and both of them are carrying the wild type allele for red eyes. The male is a white-eyed male. He's got 1 copy of the X chromosome that's carrying the mutant white-eyed allele, which we'll represent with a superscript lowercase w. But since he's a male, he's carrying 1 X and he's carrying a Y. So we can do this with a Punnett square. 1/4 of the offspring get the X with the wild type gene from mom and the X with the white-eyed allele from dad. And red eyes are dominant to white eyes, so 1/4 of the offspring are going to be red-eyed females. Actually, it's 1/2 of the offspring because another 1/4 get the other X with the red-eyed allele from mom and the white-eyed allele from dad. So half the offspring are going to be females, and all of those females will have red eyes. Half the offspring are going to be males, and 1/4 of the offspring will get a red eye, an X with a red-eyed allele from mom and a Y from dad. And the other 1/4 of the offspring will get the other X with a red-eyed allele from mom and a Y from dad. So half the offspring are going to be heterozygous females. They'll have 1 wild type X from mom and 1 white-eyed X from dad. And half the offspring are going to be males, and they will have an X with a red-eyed allele from mom and a Y from dad. But if you take 2 of those and cross them, here's what you get. 1/4 of the offspring get an X with a wild type red allele from mom, and an X with the red-eyed allele from dad. And they are homozygous. They will be normal red-eyed females. A quarter of the offspring will get X with the normal allele from dad and X with the white-eyed allele from mom. And they will be red-eyed females because red eyes are dominant to white eyes. A quarter of the offspring get the wild-type X chromosome from mom and a Y from dad. And a quarter of the offspring get the white-eyed allele on the X chromosome from mom, and they get a Y from dad, and those are the white-eyed males. So you get half of your offspring are red-eyed females, a quarter of your offspring are red-eyed males, and a quarter of the offspring are white-eyed males. The way you explain that pattern is that you have genes that are physically located on sex chromosomes, specifically on the X chromosome. And if they're carried on the X chromosome, they are probably carried on other chromosomes as well. And as we'll see, some later experiments demonstrated that that is true. Chromosomes are the physical location of whatever these gene things are. So we've finally started opening up that black box. And it turns out there's lots of examples of this other than fruit flies. Naturally, we'll go back to cats, right? I talk a lot about cat genetics just because I don't know if you all like cats, but at least you've heard of them and maybe you can identify with them better than you can pea plants or fruit flies. You might have noticed I didn't talk about orange color in cats, and the reason is that the gene for orange color is sex-linked, and it's dominant to the allele for no orange color. One odd thing that goes on is there's epistasis going on. If a cat has a gene for orange color, that will block the action of the gene for black and brown color. And that's another example of epistasis. One gene can modify what another gene does. One gene can even block the action of a second gene. That actually happens all the time. That is yet another example of epistasis. So male cats are either have an X carrying the orange allele, which means they will look orange, or X that's not carrying the orange, which I've written here as X minus, just because it's hard to tell the difference between X with a capital O and X with a lowercase o. So a male cat either has an X with the orange allele and a Y, or he has an X with the not orange allele and a Y. So male cats have to be either orange or not orange. Female cats could be homozygous. Female cats could have the genotype that will abbreviate as XOXO, in which case that female cat will be orange. Or a female cat could be homozygous for the not orange allele, and she'll have the genotype X not orange, X not orange. And then that female cat might be brown or black or gray or whatever we've talked about. But female cats can also be heterozygous. Male cats can't, because male cats normally only have one copy of the X allele. Heterozygous female cats end up with something that we can think of as being incomplete dominance. There's a little more going on than that I'm going to skip over for time reasons. But you can call this a type of incomplete dominance. A cat that's heterozygous will end up with patches of orange fur and not orange fur. That's called a tortoiseshell cat, and this cat is a tortoiseshell. I don't know if it has anything to, what exactly it might have to do with genes, but tortoiseshell cats are said to be kind of psycho. They call it tortitude. We've never had a tortoiseshell. I can't answer that from personal experience, but tortoiseshell cats have that color pattern because they're heterozygous. They've got one X with the orange allele and one Alex with the not orange allele. Okay, somebody has to leave. Toodaloo, we'll see you later. Pick this up on YouTube when you can. And incidentally, a cat that is heterozygous for orange fur and also is carrying the separate gene for white spotting is a calico cat. So that's a calico. She's got orange and not orange patches. And she's also got the completely separate allele that we talked about for white spotting. And you'll note, by the way, that torties and calicos can only be females. So there. There's some great examples of this in humans. I am assuming that most of you can see the letter or the number 12 in this picture, but some of you may not be able to. I never ask, but usually in a class this size, somebody may come up and tell me this. There's probably at least one person that cannot see the number 12 right here because they've got a specific type of color blindness called red-green color blindness. And red-green color blindness is caused by a recessive allele, and it happens to be sex-linked. Meaning that for a woman to have red-green color blindness, she must have 2 copies of the colorblind allele. But for a man to get it, he only needs 1 copy. And this is why roughly 7% of all men in the United States are red-green colorblind, but less than 0.5% of the women are red-green colorblind. And this is because the allele for color blindness is physically carried on the X chromosome. Another one in humans is the most common type of hemophilia. It's called factor IX hemophilia. That's the inability of blood to clot. If you have hemophilia, your blood clots very slowly, or it may not clot at all. And you can bleed to death from even a very minor injury that most of us would just shake off. And here's Queen Victoria of England in her later years. She was not a hemophiliac, but we've determined that she was heterozygous for hemophilia. She had 1 X chromosome with the allele for normal clotting factor, and 1 X chromosome with the allele for incorrect clotting factor. One of her X chromosomes carried the allele for hemophilia. So she must have the genotype that we could write as X. One of her X's was normal, and one of her X's had the hemophilia factor on it. Her beloved husband, Albert, by the way, being male and not being a hemophiliac, must have had a normal X and a Y. Okay, yes, Ian, I'm actually coming to that. Queen Victoria was affectionately known as the grandmother of Europe because Victoria and Albert had a bunch of kids, and they all married into various royal families. One of her sons, Duke Leopold, just happened by dumb luck to get Queen Victoria's hemophilia allele. And since he's a son, he got a Y from his dad. He didn't get his dad's normal X chromosome because if he had, he wouldn't be male. He would have been a she. Leopold was hemophiliac. He married a normal female, and their daughter Alice inherited his hemophilia allele and a normal allele from mom. So his daughter Alice was heterozygous, and she passed it on to Viscount Tremonton over here, who just by luck ended up with the hemophilia allele. And because he was a male, he must have gotten a Y chromosome. And so he died young of the effects of hemophilia. Now, Edward VII is the ancestor of the current ruling family of England, and his family never had hemophilia. So he must have inherited Queen Victoria's normal allele and a Y chromosome from Prince Albert. So he never had the hemophilia allele and could not pass it on. So the present British royal family does not have hemophilia in it. Edward VII was the father of George V, who was father of George VI, who was father of Queen Elizabeth, who's on the throne now. And she, of course, is the mother of Prince Charles, who's the father of William and Harry. One of them married Meghan Markle, and you know the rest. But there's no hemophilia in the present royal family. But Queen Victoria's daughter Beatrice over here, she inherited a normal allele from dad because he had X plus Y. He had a normal X chromosome and a hemophilia allele from mom. She married into the royal family of Spain, and two of her sons had hemophilia. Just by luck, they got her hemophilia X chromosome. Her daughter Victoria inherited a normal X from her father, and she just so happened to get the hemophilia chromosome from mom. Whoops. And she passed it on to two dukes of the Spanish royal family, Alfonso and Gonzalo down here, who both had the X chromosome for hemophilia and a normal Y. And they died young because they were not only hemophiliac, they were into racing fast cars. And that's not a very good combination. They both bled to death after car accidents. And then, as Ian brought up, Queen Victoria's daughter Alice was a carrier. She had a normal X chromosome from her dad and a hemophilia chromosome from her mom. And she passed on the hemophilia chromosome to Alexandra right here. And Alexandra married Tsar Nikolai II of Russia. And Alexandra passed it on to her daughters. There were four, Olga and Tatiana and Alexandra and Anastasia. And she passed it on to her son, Alexei, right down here, who just happened to get Queen Victoria's hemophilia and a Y chromosome because he was male. He was a hemophiliac. We don't know if any of the daughters could have been carrying hemophilia because they were all murdered in 1920 by the communists. But Crown Prince Alexei down here was a hemophiliac because he got it from his great-grandmother, Queen Victoria. So there's a couple of pictures of people. Okay, the third was Maria, thank you, not Alexandra. Olga, Tatiana, Maria, Anastasia, and Alexei. Anastasia ended up as the heroine of that animated movie some of you might have seen, Anastasia, because there were rumors that she survived. Unfortunately, they're not true. They found her remains. But it made a nice movie. Anyway, Queen Victoria's granddaughter, Victoria Eugenie, was a carrier, and she's the one who passed it on to the Spanish royal family, although the current King of Spain doesn't have it. And then her granddaughter, The Last Zod on Netflix. I have not seen that. Very informative. Cool. Victoria's granddaughter, Alexandra, married Tsar Nikolai. There they are. Nikolai sitting. Alexandra is right there. She's the hemophiliac character. And Anastasia's the youngest of the girls right here. That's her. And the others, I can't remember which one is which. But Anastasia's got her arm right there around Alexei, the Tsarevich, the crown prince right there. He's the one who would have become the next emperor if the communists had not revolted in 1917. And he was a hemophiliac because Queen Victoria was his great-grandma. And it just so happened, just by dumb luck of the draw, that he had inherited her X chromosome that was carrying that hemophilia allele. Minor historical note, desperate to help her son, Alexandra found a mystic and faith healer who did seem to help Alexei. It might have been the placebo effect, who knows, but he did seem to be able to help him a bit. He became very close to the royal family. There were rumors that he had an affair with Alexandra, although I don't think anybody really knows. I'm not sure if historians believe it, but the rumors are there. And thanks to those rumors, he has been immortalized as Rasputin, lover of the Russian queen. It was incredible how he carried on. No. Rasputin, Russia's greatest love machine. It was incredible how he carried on. Anyway, that is, I believe it's Rasputin, not Rasputin, but that's him. So the reason, if it had not been for Queen Victoria's hemophilia showing up in great-grandson Alexei, we would never have had that pop hit. Rasputin, lover of the Russian queen. Okay, in the animated film, he's an evil wizard. There's all sorts of legends about him. I have no idea if they are true or not. There was a lot of slander spoken about him because people resented the way he had access to the royal family, which basically meant ultimate power in Russia. I doubt anyone will ever know the truth because the people that did all ended up shot. The royal family was executed in 1920, and unfortunately, we now know they did get Anastasia. Okay, anyway, so there's your little interaction between genetics and history, right? Knowledge is all one. We can make links between biology and all sorts of other fields that you would think would be separate. Anyway, stepping away from the Russian royal family, Morgan and his students, Bridges and Sturtevant, ended up winning the Nobel Prize in, I think, the 1920s for demonstrating that genes are also carried on autosomes, which means all the chromosomes that are not sex chromosomes. And Sturtevant was able to use a really nifty trick that I don't really have time to explain in detail. You can look it up if you really want to know. It's called linkage mapping. There's a way you might remember that when chromosomes pair up in meiosis 1, they break and they do what we call recombining. They will pair up, they will break at particular points, and then each chromosome ends up with a piece of the opposite chromosome attached to it. It would be like me standing next to one of you and cutting off and swapping our left arms. Like if I ended up with your left arm and you ended up with mine, that's weird if it's us doing it, but it's what chromosomes do all the time. They pair up and they swap equivalent pieces. Sturdivant was able to work out how frequently this happens when you do a cross, and was able to use that to map the location of genes on chromosomes. And so he's able to work out where on chromosomes genes are physically located, and was able to use that to construct what we call linkage maps. What you're looking at here is a partial linkage map of the fruit fly X chromosome. The numbers that you see on the left represent the location of each gene on the chromosome, and they're in units called centimorgans, after T.H. Morgan. And then on the right, you can see the names of the different traits: yellow body, scute bristles, white eyes, facet eyes, ruby eyes, cut wings, singed bristles, and so on. One of the things you might notice is that none of these genes that are listed here seem to have anything to do with development of the sex organs. And that's because the X chromosome in fruit flies and also in humans has a lot of genes on it that have nothing to do with your gonads, nothing to do with your plumbing. In humans, the gene for, okay, we've already talked about the gene for factor IX blood clotting function is carried on the X chromosome, and everybody needs that, male or female. That's got nothing to do with your sex or your gender. The gene for the protein in your eye that makes it possible for you to tell red and green apart has nothing to do with your sex, but it's carried on the X chromosome. There are lots of very important genes that nobody can live without that are carried on the X chromosome. And that's part of the reason why you don't have anybody with the YY chromosome type. If you could come up with that, there's no way that person could live. An egg with two Y chromosomes would miscarry very quickly. You can't do without an X. Okay, somebody just typing in a name. In humans and in fruit flies, the Y chromosome is really small, and there are very few genes that are physically carried on it. In humans, one of the few Y sex-linked genes is a gene for hair in your ears. There are people that grow, like, we're not talking about light fuzz, we're talking about great big bushes of hair growing out of their ears. You know, it looks like an armpit in their ears. I'm not even kidding. That's about how long the hair can get. And if a man has that trait, he will pass it to all of his sons and none of his daughters. And none of his daughters will ever have offspring that have big bushes of hair out their ears. But all of his sons will, and all of their sons will. That's a fairly rare case of a gene that has nothing to do with sex that is linked on the Y. But there's lots of genes that have nothing to do with your sex that are linked on the X. The main thing that your X chromosome does, or your Y chromosome does, sorry, if you've got one, is it's got a region on it called the SRY. And SRY acts as a, you could think of it as being a switch that is thrown when you are about the size of a bean. Every embryo, every developing, soon to be baby starts out life with the same basic pattern of plumbing. And I wish I had time to go into this in a little bit more detail, but it's a fascinating story. Every embryo starts out with genitals that look more or less like the female pattern. With an opening, a little knob of tissue that might eventually end up as the clitoris, and flaps of tissue around the opening, similar to the labia. I'm assuming everybody knows what genitals look like by the time you're at this age, because I don't have diagrams handy. Everybody looks more or less like that. And everybody's got two pairs of tubes internally that connect to a lump of tissue called the Wolffian body. If you have a Y chromosome, one pair of those tubes becomes the spermatic cords, and the other pair of tubes doesn't become much of anything. That lump of tissue drops down and becomes your testes, and your external genitalia change. That tiny nub of tissue grows out and becomes your penis. Your urethra moves position so it ends up at the tip, and those flaps of tissue fuse and form a scrotum into which your testicles descend, and you end up with the male phenotype. I'm assuming everybody knows what that looks like. If you don't have a Y chromosome, the other pair of tubes fuses together and becomes a uterus. That lump of tissue stays inside and becomes your ovaries, and the little nub of tissue and the flaps of tissue stay pretty much the way they are, and they become a clitoris and labia. So both male reproductive systems and female reproductive systems are basically built from the same parts. And what the Y chromosome does is it's got the switch that triggers them to develop towards the male typical pattern rather than a female pattern. If that switch is not working, you can end up with people, it's kind of rare, but it does happen. You can end up with people who are XY and female. You can end up with people that are kind of halfway with ambiguous genitalia. There's one population of people in, I think it's Guatemala, where for interesting reasons having to do with hormones, the transition from female to male anatomy does not happen when people are still embryos. It happens when they hit puberty. They have a condition called huevadose, which is Spanish more or less for getting your balls at 12. It's really cool. They look female when they're born. They look female until they hit puberty, and then spring out sprouts a dong, which rather complicates dating, I suppose. Hey, Mom, you know that prom dress? Can we swap it? Yeah, it's really cool stuff, but that's all the Y chromosome does. Except for a few minor genes, it's got the switch that makes your anatomy develop in the male pattern instead of in the female pattern. It is the Y chromosome that is responsible for giving you a tallywhacker and not a hoo-ha. Hope you got my technical terminology there. Okay. So that's important, and that won Morgan and his students a Nobel Prize, which is kind of a big deal. A pivotal discovery that genes are physically carried on chromosomes, and they're actually carried in an order that we can work out. We still don't really know what chromosomes are or what they do, but we can work out that genes are on them somehow. Now can we open that black box anymore? We shift the scene. London, 1928. Now we'll do it in Golden Girls style. Picture this, London, 1928. A British bacteriologist named Frederick Griffith has been studying the bacterium that causes bacterial pneumonia. You can get pneumonia because of viruses. I have not yet covered what viruses are. I will get to that soon, but I can't yet talk about it because we've still got some background material I've got to plow through. But you can also get pneumonia, lung infections, from a bacterium called Streptococcus pneumoniae. Obviously, this is something that you need to study if you want to develop treatments against it, which is what Dr. Griffith was working on. This is Streptococcus pneumoniae. Strepto means that they come in chains. These chains happen to be really small. And these bacteria have what look like halos around them when you stain them and look at them under a microscope. And the reason is that those cells are surrounded by a capsule of snot, of this mucus-like material. You might remember I brought this up way back at the beginning of class when I was talking about bacteria. Some bacteria have a capsule. This is why I brought that up in the first place. The reason that matters is that bacteria with the capsule have armor against your body's immune defenses. So Streptococcus pneumoniae bacteria that have that capsule can cause severe disease. They're protected against your body's ability to fight them off. Bacteria that don't have the capsule, your immune system can usually beat. So bacteria without the capsule might make you briefly sick, but they're much less likely to cause severe disease because they're not protected. Okay. I don't think you're going to get to do this lab, but we're able to do it in normal years. If you're studying bacteria, you can spread them out in a glass dish that's been filled with a gel-like substance. It's roughly got the texture of Jell-O. It's actually a carbohydrate that's extracted from certain seaweeds, and it's called agar, A-G-A-R. And you can mix nutrients in the agar so your bacteria feed. They'll grow on top of the agar, but they don't eat the agar. Well, there's a few that can, but most bacteria can't break down agar, but they can absorb nutrients from it. So people that study bacteria grow a lot of bacteria on agar plates. And if you put one bacterium on an agar plate, it will grow and divide and become 2, 4, 8, 16, ... until after a day or two, you end up with a big clump of bacteria called a colony. So each one of these little dots things right there is a clump of thousands, maybe millions of bacteria all packed together. And pneumonia bacteria that don't make that capsule form colonies that look kind of small and rough, and they're called rough type or type R. Streptococcus pneumoniae that does have the capsule grows in colonies that are a lot bigger and a lot shinier because the bacteria are making that capsule of snot. And we call this S-type bacteria, S standing for smooth. I promise this is going to be relevant real quick. R-type bacteria don't have the capsule, and R-type bacteria don't cause serious disease. If you breathed in some R bacteria, you might get a bit of a cough, but your body can fight those off easily. If you breathed in smooth type bacteria into your lungs, you could end up with severe lung disease. And recovery might take a long time. Here's the weird thing. This is the last thing we'll talk about. You can test these bacteria by injecting them directly into mice. Yes, perhaps not pleasant that we experiment on animals, but sometimes there's no other way to find these things out. If you inject, S-type bacteria, those smooth types with the capsule, directly into a mouse, the mouse dies within 24 to 36 hours of massive infection. And then if you take samples of that mouse's blood and you spread them on a dish of agar, you get live S bacteria back. So you inject S into the mouse, the S bacteria multiply very fast inside the mouse, the mouse dies of infection, and the dead mouse's blood contains S-type bacteria. Okay, that's not surprising at all. Inject R bacteria into a mouse, and the mouse stays healthy because the mouse is able to fight off infection with R-type bacteria. So live S kills the mouse, live R doesn't kill the mouse, dead S doesn't kill the mouse. Griffith found that you could take S-type bacteria that he'd killed by heating, inject them into the mouse, and nothing happens, right? Dead bacteria are just mopped up by the body and they don't cause any harm. So he mixed a mixture of live R and dead S and found, much to his surprise, that holy cow, that did kill the mouse. Live R bacteria, the mouse fights those off. Dead S bacteria can't grow because they're dead. Neither one of those will kill the mouse if you inject them separately. Mix them together and it kills the mouse, and the mouse are found to have living S bacteria in their bodies. That's freaking scary. Griffith reasoned the only way that could happen would be that if the dead S bacteria, they might be dead, but they still contain something that could be picked up by the live R bacteria and turned them into live S bacteria. Something had gone from the dead S bacteria into the live R bacteria and turned them into live S bacteria. Something went from the dead to the living. This is like a zombie movie. And whatever that was transformed the living bacteria from a harmless type to a harmful type. He didn't know what that was. He just called it the transforming principle. But what exactly is this? This is just a name so far. What is the transforming principle? Well, an American named Oswald Avery and a bunch of colleagues of his started growing lots of Streptococcus bacteria, breaking them down chemically, taking different substances out of them, and testing which one might be the transforming principle. So you might grind up a bunch of bacteria, break them down into the molecules that they're made of, and start testing each molecule to see if it has the effect that Griffith had noticed. It took them 15 years, but they finally isolated the transforming principle. It turned out that it was a molecule that had already been discovered back in 1869 by a guy named Miescher, a Swiss guy, I think. It turns out that the transforming principle was present in eukaryotes as well as prokaryotes. In eukaryotes, it was only present in the nucleus of eukaryotic cells. And the substance was also a very weak acid, and it happens to be called nucleic acid because it's an acid and it's found in the nucleus. Duh, there we go. And it turns out that nucleic acid is one of the substances that chromosomes are physically made of. The other substance is called protein. You might have heard of proteins, especially if you know about diet and nutrition. We'll get to what proteins are later, but Avery was able to show that protein is not the transforming principle. Chromosomes are made of protein and nucleic acid. This picture, by the way, you can isolate nucleic acid. We used to do it in this class by taking tissue. We'd use strawberries, freezing it, grinding it up, breaking up the cells using strong detergent, and then getting the nucleic acid out using alcohol. So you can extract nucleic acid in your kitchen sink using just dishwashing liquid and Everclear, if you ever want to. And nucleic acid is this kind of pinkish cotton candy looking stuff that's floating dead in the center of the test tube. That's what nucleic acid looks like if you extract it from cells. So nucleic acid is the transforming principle in bacteria, and it's one of the substances that chromosomes are made of, so it's present in eukaryotes as well. So figuring out how chromosome can actually carry genes is going to depend on finding out more about what that transforming principle is, and that's what we will cover this coming Tuesday. But for now, we will sign off because it's the end of lecture, and I'll see you on Tuesday, same bat time, same bat channel, as they used to say on the old Batman TV show.